Good morning, everyone. Welcome back in my YouTube channel, Microbiome 2020. Today I will explain the topic epinephrine cascade. Model for epinephrine cascade. We know about the epinephrine. Epinephrine is a ligand bind to the receptor adrenergic. The binding of epinephrine to the site on the receptor deep within the plasma membrane that promotes the conformational changes in the receptor. That is the intracellular domain that affects its interaction with an associated G protein and promoting the dissociation of GTP and binding the GTP from the cytosol. We know about that the beta adrenergic pathway in which the epinephrine is used. Okay, this is the model for the epinephrine cascade. In this, the epinephrine triggers the series of reaction in hepatocyte in which the catalyst activates the catalyst. This catalyst activates the catalyst. Again, this catalyst activates this catalyst. Again, this catalyst activates another catalyst. That is, epinephrine triggers the series of reaction in hepatocyte in which the catalyst activates the catalyst, resulting in the amplification of original hormone signal. That is the amplification of original hormone signal, means the transmission of message from cell surface with the help of amplification. The number of molecules shown are simply to illustrate the how to work amplification and our amplification and number of number of molecules activated, which are the number of molecules activated. See, the binding of one molecule of epinephrine to the 1-beta adrenergic receptor on the cell surface. Okay, that activates the many, that is possibly the hundreds of G proteins, and one after another. Each of this G protein which goes on to activate the adenylyl cyclase. Each of these G protein goes on to activate the adenylyl cyclase, and this one molecule of adenylyl cyclase acts catalytically and producing the molecules of cyclic AMP from ATP. Okay. Then these two molecules of cyclic AMP are required to activate the one molecule of protein kinase A catalytic subunit. I have already told about the epinephrine triggers the reaction, catalyst activates the catalyst. Okay, this cyclic AMP activates the protein kinase A. Again, this protein kinase A activates the phosphorylase kinase. Then again, this phosphorylase kinase activates the phosphorylase B, and then from this phosphorylase B, molecule of phosphorylase B activates and converts the glucose from glycogen. This is the process of amplification, also model for the epinephrine cascade. Epinephrine binds to the one molecule of epinephrine binds to the receptor, then activate the G protein, then activate the adenylyl cyclase. This adenylyl cyclase activate the cyclic AMP, then cyclic AMP again used for the activation of the protein kinase A. Then protein kinase A activates the phosphorylase kinase, and phosphorylase kinase the phosphorylase kinase B. Activate the phosphorylase B, and from this phosphorylase B, the production of the glucose from the glycogen. This is about the model of epinephrine cascade. Next one is the cascade, that is amplification, means which transduction step activates the how many molecules, number of molecule. See here, it is the enzyme. In enzyme cascade, its system amplifies the biological signal. A very small number of polar molecule makes a choice to trigger the production of enzyme molecule that catalyze the reaction tens over the hundred of substrate molecule. For example, a single molecule of epinephrine binds to the receptor, one molecule of receptor form the epinephrine receptor complex and activated and trigger the adenylyl cyclase, activated and trigger the adenylyl cyclase with the help of G protein in a cytosol of the plasma membrane. Okay, then this adenylyl cyclase makes the catalyze the information of 100 molecules of cyclic AMP from ATP. Okay, this adenylyl cyclase makes catalyze the information of the 100 molecules of cyclic AMP from ATP. Then each of these cyclic AMP can activate and can catalyze the molecule and can catalyze and reactivation of molecule protein kinase A. Okay, then same, each of these protein kinase molecule make catalyze the reactivation of 100 molecule of phosphorylase B kinase. Got it? From protein kinase A, each molecule of protein kinase A make catalyze the reactivation of the phosphorylase, 100 molecules of the phosphorylase B kinase. Then from these, each of those molecule make catalyze the reactivation of 100 molecules of glycogen phosphorylase A. Glycogen phosphorylase A. And then each of those molecules make catalyze and conversion of the 100 molecules of glycogen to glucose 1 phosphate. What? Each of those molecule makes catalyzed and conversion of glycogen to the glucose 1 phosphate, and this glucose 1 phosphate, that is glucose, transferred to the blood. This is the cascading, epinephrine cascade, and that of the 100 million of the glucose molecules are the response of each molecule of epinephrine. Means one molecule of epinephrine carry the signal transduction process as an amplification, and step by step activates the catalyst. Catalyst activates the catalyst, and response is a number of molecules of glucose. Got it? This is the epinephrine cascade. Then this whole figure shows the outline of epinephrine cascade, and each transduction step activates the number of molecules, and which is the response of the epinephrine cascade. This is the first primary step, reception, how to bind up epinephrine to G protein, one molecule. Then transduction procedure carried step by step, and response obtained as a glucose 1 phosphate about the 10 raised to 8 molecules. This is all about the outline of epinephrine cascade. Then summary of this cascade: the enzyme cascades in which the single molecule of hormone activates the catalyst to activate the another catalyst, and process carried and so on result in the large signal amplification that is characteristics of hormone receptor system. Got it? Thank you. Now I will stop here.